Welcome to EduRev.
Welcome students. I'm Claris Pereira Colasso and I have been training NEET medical aspirants in botany and zoology as well as NCERT based board exams for the past 8 years. In the previous two video lectures, we have already studied a little bit about pre-fertilization structures and events with respect to the androecium, the male reproductive whorl of the flower, and we have also studied the pre-fertilization structures of the gynoecium. In today's video lecture, we will be focusing on the pre-fertilization events of the gynoecium, so that is none other than megasporogenesis, and we will also be studying another event, and that is pollination. That is, what are the various types of pollination available for a plant. So let us quickly revise through the structure of the embryo sac or the female gametophyte. If you remember, we called it as the female gametophyte simply because it houses the female gamete, which is the egg cell. So we said that the embryo sac consists of 7 cells but 8 nucleated, which is important for us to remember. Okay, so here you have the presence of antipodals, so 3 cells in total, 1 central cell. Okay, so if you remember, the central cell was formed by the fusion of 2 polar nuclei. If we consider the 2 polar nuclei, they're considered to be haploid in nature. If we talk about the central cell, since it's a fusion product, we come up with a secondary nucleus, but this is the diploid cell of the embryo sac. And finally, we have the egg apparatus, which contains 3 cells in total, 1 egg cell and 2 synergids. So for our revision's sake, if you remember, antipodals are always towards the chaliceal end, your synergids, your egg apparatus along with the egg cell always towards the micropylar end, and the polar nuclei or the central cell present more or less in the center of the embryo sac. Also, for our revision purpose, we must recall that the chaliceal end is actually the base of the embryo sac or the base of the ovule, and the micropylar end is the top of the ovule. Okay, remember, we're talking about an anatropous ovule, which is an inverted ovule. Okay, so we need to remember these particular points as we enter into megasporogenesis. So megasporogenesis is nothing but the process by which we are able to form a megaspore. Now we must remember that the megaspore is essentially not really the egg cell, but the megaspore will result in the formation of the egg cell. Likewise, if you remember, we have already studied microsporogenesis, and microsporogenesis was required to form the microspore. So the pollen mother cell gave rise to the microspore. But the microspore was simply the pollen grain. It was not the gamete. It housed the gamete within. So let's start with megasporogenesis. Now, in order to understand megasporogenesis, we must recall that in the ovule of the plant, we have the presence of a lot of nutritive cells called as the nucellus cells. So very clearly in our figure, we can see a whole lot of nucellus cells there. However, one of these nucellus cells, just one, not all, okay? So just one of these nucellus cells starts becoming a little more specialized as compared to the others. Now, what do we imply by becoming a little more specialized as compared to the others? It starts retaining a lot of fluid. In other words, it is beginning more or less to undergo an interphase-like event. It is starting to double its cytoplasm. So you see this particular event taking place in just one nucellus cell. And this particular one nucellus cell that becomes more specialized than the others is going to be called as the megaspore mother cell, or the MMC, which is the meiocyte in this particular case. Now I want you to remember, when we spoke about microsporogenesis, that is the formation of the pollen grain, we said that all sporogenous tissue, each and every one of those cells, behaved as a potential mother cell, which means all of them were meiocytes. However, in this particular case, all the nucellus cells do not behave as meiocytes, just one of them becomes more specialized in order to become a meiocyte. So that is important for us to understand. Now, what really happens to this one MMC? MMC has already undergone an interphase-like event, so it obviously has to enter into meiosis. Now, it enters into meiosis 1, which is none other than reductional division, and very clearly you can observe the presence of a Megaspore diet, which means one cell, a diploid cell, has divided to give you 2 haploid cells. That's the Megaspore diet. Now, meiosis event has not yet been completed, so therefore you undergo meiosis 2. And in meiosis 2, you can very clearly observe that the Megaspore diet is now going to be converted into the Megaspore tetrad, which means you get 4 haploid cells. Now, if you are able to recall, when we spoke about pollen grain formation, all the 4 microspores that were formed, the microspore tetrad, all the 4 microspores were equally going to behave as viable pollen grain. They all had the same capacity to continue to grow and become viable pollen grain. However, there's a small difference in case of the female reproductive system. Now here you will find out of the 4 megaspores that you get, so a megaspore tetrad, just one of them is going to survive and 3 of them are going to degenerate. And it is that particular one of those 4 that survive that gives rise to the embryo sac eventually. Okay, so therefore in this particular case, the entire embryo sac is formed from just one functional megaspore, the remaining 3 die off, and this is therefore called as monosphoric development. Now we need to understand why is such an event taking place in the female reproductive system but not really occurring in the male. The first reason is, remember, in the male you obviously require a whole lot of pollen so as to make sure that you have some amount of pollen reaching the stigma. So it is important to get numerous pollen in the anther. But remember, as we go into higher organisms, let's take for example human beings. You will obviously see that in human beings, when a mother conceives, it is most often that she conceives just one offspring at a time, one zygote at a time, one baby at a time. Now the reason is this: a lot of nutrition goes into that one, a lot of care goes into that one. So in higher organisms, one is generally preferred over having multiples. Not to say that you will not have multiples, but this is a general trend. So therefore, here also you will find that in the female reproductive part, that's the gynoecium, out of the 4 out of the 4 megaspores that you have formed, one of them remains functional, 3 of them will degenerate. Now let's try to understand what happens to that one functional megaspore and how does it result in the formation of the embryo sac. Now, in order to give rise to the embryo sac, you have 3 different events that must take place. The first event that must take place is the presence of mitotic divisions. But these events of mitotic divisions are not simple mitotic divisions. Okay, so you have the first, the second, and the third mitotic division, but they're not just normal divisions. Now, if you recall your cell cycle, after you have your interphase, it is important that you have karyokinesis, that's the division of the nucleus, and immediately followed by cytokinesis, that is the division of the cytoplasm, and if required, cell wall to be formed, for example in plant cells, which we require here. However, there can be mitosis in which you simply have interphase followed by karyokinesis and then no cytokinesis. So interphase, karyokinesis, and then you can revert back into another interphase and then again into karyokinesis. Now eventually what happens in this particular case is you keep on increasing the number of nuclei since karyokinesis is going on, and you keep on doubling the amount of cytoplasm since interphase is going on. However, there is no cell wall formation. So you have a whole lot of cytoplasm. So say suppose you had a small cell initially, you will find that the cytoplasm in that cell will become really huge. So you have bulk amount of cytoplasm, and you'll find that the nucleus, instead of remaining a single nucleus, you'll have a multinucleated condition. Now such type of division is called as free nuclear division. So you will find that those 3 mitotic divisions, those 3 events that take place, are all going to be free nuclear divisions. Now after 3 mitotic divisions take place, whatever nuclei are present in that one single cell will then undergo cytokinesis, will then undergo cell wall formation, and that particular event is going to be called as cellular division, formation of cells. Okay, so 3 mitotic divisions, which are all free nuclear divisions, followed by a cellular division. Now how does that really occur? So one of the megaspores of the megaspore tetrad will undergo the first mitotic division. So you can see the amount of cytoplasm. Now remember, this is a single cell. The amount of cytoplasm increases and the nuclei doubles. So single nucleus undergoing one mitosis, you will have 2 nuclei. Now the 2 nucleated embryo sac as such will undergo the second mitotic division. So each of those nuclei are going to double. Okay, so in total you will result in a 4 nucleated condition. And if you have the third mitosis taking place, those 4 nuclei are going to double. So you'll have an 8 nucleated condition. So till here you have a free nuclear type of division that's going to take place. Absolutely no cytokinesis. But once you have 8 nuclei, now cellular division takes place, which means these 8 nuclei are going to arrange themselves within that whole, actually it's just one cell that has elongated, so within that area, okay? And this is how you're going to result in the formation of an embryo sac. Now, if you clearly visualize, we have 8 nuclei at the moment. The moment you start forming cell walls, you will get the embryo sac as we know it. Okay, so remember, 3 mitotic divisions were completed, free nuclear division, all those 3 mitotic divisions were free nuclear, and then finally now we're going to look at what the cellular division looks like. Okay, so if you look at the cellular division, you will see 3 nuclei pushed towards the chaliceal end. Okay, all of them separated with a cell wall. You'll find 2 nuclei in the center covered with a cell wall, so that's cellular division taking place there, or rather formation of a cell wall. And then you have the synergids down there along with the egg cell. So very clearly, after you had 3 mitotic free nuclear divisions, you start having cellular divisions. And here you find that you have an entire mature embryo sac as we know it. Now let's move into the next part, and that is pollination. Now there are various kinds of pollination, and we can classify them into two, that is self-pollination and cross-pollination in case of plants. Now, self-pollination can also be of two different types. One is called as autogamy, auto implying same. The second is Xenogamy, and cross-pollination is of only one type, or you just have an alternative term there, and that is called as Xenogamy. Okay, so let's try to understand these terms in depth. Starting with the word autogamy. Autogamy is a type of self-pollination in which pollen is transferred from the anther to the stigma of the same flower. However, Xenogamy is the transfer of pollen from the anther of one flower to the stigma of another flower, but both these two flowers belong to the same plant. Now, even though they're two different flowers, but they belong to the same plant, there is no genetic variation between those flowers ultimately. So therefore, we place autogamy and Xenogamy under self-pollination. However, cross-pollination called as Xenogamy, Xeno gives you the understanding of something different. For example, xenografts are grafting between, let's say, humans and animals. Okay, so those are called as xenografts. So xeno implies something absolutely different. Now, let's start with the first type of pollination, that is autogamous pollination. So autogamy is the transfer of pollen from the anther to the stigma, however, belonging to the same flower. Now, autogamy is a very rare event, or rather it's very difficult to get autogamy, simply because two prerequisites must be satisfied. Now, what are those prerequisites? The first, there must be synchrony between the sexes. Now, what do we imply by that? It basically means that when the anther dehisces, the stigma should become receptive at the same time. So, both events must take place at the same time. Synchrony is required. The next is, you require proximity between the sexes. For example, the anther and the stigma cannot be very far apart. They must be close by so that you can ensure autogamy can take place. Now, this would also imply, suppose we do not want self-pollination, we do not want autogamy. What is the best way to get rid of it? What could be some of the ways? Now, those are not the only ways. What could be some of the ways? Number one, probably allow the stigma to become receptive at a different time from which the anther is dehiscing, so don't have synchrony. The second, avoid proximity. Keep the anther on one side, keep the stigma on the other side. So that would prevent self-pollination, that would prevent autogamy. Okay. Now, there are a variety of different types of autogamous plants. Okay, now for example, three particular plants, oxalis, commelina, and viola. These particular plants have two types of flowers within them. One flower is called as a chasmogamous flower. It's a normal open flower. And the second type of flower is called as a cleistogamous flower. Now, cleisto implying it is closed. Let's have a look at these two flowers. So here you have the chasmogamous flower. Very clearly you can see that it is an open flower, and since it is an open flower, you can obviously understand, yes, self-pollination may be permitted, but cross-pollination is also possible here. So it is not simply stuck to self-pollination. Self or cross is also possible. However, let's look at the cleistogamous flower. Now the word cleisto means closed. Now this particular flower looks like a small bud. Actually it's a mature flower, but it's closed. Now since this particular flower is closed, we can understand that when the anther dehisces, whatever pollen is present there is going to fall onto the stigma of the very same flower. There's absolutely no movement. Okay. Now there's some advantages of having such type of a pollination. The first thing is, I'm sure pollination will occur. So there's assured pollination. Now assured pollination also assures me fertilization, also assures me that I will get some seeds. Okay, so there's assurance of seeds in this particular case. And the second is you will have purity of the trait that you have. For example, if it's a homozygous plant, it will continue remaining homozygous. Okay, so that is really important sometimes when we have breeding programs. We want to maintain purity of the breed. Okay, so those are the advantages. However, there are a number of disadvantages to this particular type of Cleistogamous autogamy pollination. Now, the first disadvantage is this: you always have self-breeding. And when you continuously have self-pollination or self-breeding, it will result in something called as inbreeding depression. Now, inbreeding depression is a concept or is an understanding in which the offspring start losing their viability to have further offspring, which means, in other words, offspring start becoming sterile. Okay, now this is obviously not what we want. We want the generation to continue. So inbreeding depression is a problem if you're going to have constant self-pollination or self-breeding. The second is there's lack of variation. Now when there's lack of variation, if you start having changes in the climate, environmental changes, you will have absolutely no capacity to adapt. So adaptation is avoided or is almost nil in this particular case. So that is Cleistogamous flowers. Now let's move into Geitonogamy. So Geitonogamy is the transfer of pollen from the anther to the stigma, but remember, of different flowers. However, both flowers belong to the same plant. Now, Geitonogamy essentially is said to resemble both self and cross-pollination. Now, what do we mean by that? Why does it resemble self-pollination? It resembles self-pollination genetically in the sense there's absolutely no variation coming. However, it resembles cross-pollination in the sense that you do require a pollinating agent, maybe wind, water, or maybe some insects. So functionally, it is like cross-pollination. You require transfer of the pollen grain. Okay, so nothing much to study under Geitonogamy. Let's focus on the third one, and that is Xenogamy. Now, Xenogamy is also called as Aloegamma. If you remember, your chromosomes are called autosomes and allosomes. The word allo means different. Okay, so when we talk about allogamy, allogamy is obviously transfer of pollen from the anther to the stigma of two different flowers, and both of them belonging to, say, two different plants. Okay, so two different flowers, two different plants. Now the main advantage over here is you have a whole lot of variations, helps you in adaptation, helps you to survive for a longer period of time. Okay, so the ideal way of pollinating is always going to be cross-pollination. Now, since the ideal way of pollination is always going to be cross, obviously inbreeding is not preferred and outbreeding would be preferred. Outbreeding meaning with other plants, cross. Okay, so outbreeding devices are of 4 different types. Now one can think, what is the most obvious way in which I can force cross-pollination? Now in case of plants, the most obvious way to force cross-pollination is have a unisexual plant, have unisexual flowers. So if plants are unisexual, you will definitely have a separate male, a separate female. You will be forced to have cross-pollination. But suppose it is not possible for the plant to have unisexual plants, okay, that species, sorry, to have unisexual plants. Then suppose if bisexual is a must, then what can we do? We can avoid synchrony of the sexes, okay, synchronization of the receptivity of the stigma and the anther-dehiscence can be avoided, so lack of synchrony. Now, suppose this also is not going to be possible. You cannot avoid synchrony. Synchrony does take place in that particular species. Then the next thing that could possibly be done is place the anther and the stigma away from each other. Place these whorls, the androecium and the gynoecium, separately, different, far away from each other. Okay. Now, let's assume that that also is not possible. And both of them are present very close together, which means there's a very high tendency to have self-pollination. Now, the ultimate thing that can be done is self-incompatibility. Now, self-incompatibility refers to the fact that self-pollen falls onto your stigma, and that particular stigma does not allow the germination of your self-pollen grain, which means if a stigma has self-pollen and cross-pollen, it will allow the cross-pollen to germinate and form the pollen tube, but it will prevent the self-pollen from forming its pollen tube. So these are some of the outbreeding devices that you must remember. Now let's go through a couple of MCQs to help you out with your NEET exam. The first, cross-pollinating plants. So we have got a couple of options. We'll have to choose which ones are correct. Okay. So cross-pollinating plants, number one, produce offspring having variations amongst themselves. Now, this is obviously a correct statement. Okay. Number two, it reduces evolutionary stagnation of the population, which basically implies we have variations coming in. There's no stagnation of the genes. Okay, this is also correct. Let's go to the third one. It results in the production of pure line homozygous offspring. But this is false. This is what you see in autogammy, not in xenogamy or cross-pollination. Number four, having bisexual flowers or monoecious condition. Now, cross-pollination, you would prefer having unisexual flowers. Okay. So if you look at the options here, the correct option here would be option A, which means statement 1 and statement 2 are correct. Let's go to the next question. Aloegammy is useful because it results in dash. Option A, formation of seeds, but seeds will anyhow be formed irrespective of pollination, so that's wrong. Formation of parthenocarpic fruit, so parthenocarpy implies a fruit without a seed that we are not really sure of, so let's just wait with that particular option. Option C, formation of superior progeny. Now, allogammy results in variation, so most likely that's the answer. Option D, formation of male offspring. Now, you cannot be certain that you're only going to get a male plant, so therefore the answer is going to be option C. The next, the number of nuclei in a fully developed male and female gametophytes are dash respectively. Now, obviously, the male gametophyte, before we look at the options, we can think about it. The male gametophyte has one vegetative cell, so the pollen grain has one vegetative cell and has got two male gametes, which means the male gametophyte has got 3 nuclei. Female gametophyte, we know once if the pollen nuclei have fused, you have 7 nuclei. Okay, if they are unfused, you have 8 nuclei. But remember, mature embryo sac, so 7 nuclei. So you could have looked at the options here. Option A says 2 and 8. That's definitely wrong. Option B says 3 and 8. That could probably be right. 2 and 7, that's wrong. And option D is 3 and 7. So your answer is between B and D. But very clearly, D will tell you that you have a mature embryo sac, and that's the correct option here. The fourth question. Monosporic development describes one spore develops into a pollen grain. Now, monosporic development is with respect to the female, so no question of pollen grains here. One gamete of the pollen grain fuses with one egg cell, that's false. One megaspore is functional and the other 3 degenerate. Now, this is a true statement, but this is not monosporic development. Option D, embryo sac formation from one functional megaspore. Now, that is what we mean by monosporic development. Okay, the formation of the embryo sac from that one functional megaspore. So students, I hope you have understood megasporogenesis and various types of pollination that we have studied. I request you to go through EduRev Infinity, where you will come across a whole lot of question papers, other videos from NEET experts, and you can go through the notes as well. I'm sure this will help you in your nationwide ranking. Thank you, students.